Hello everyone. So today we begins with the practical of platelet count. What is the principle of this practical? As normal platelet count runs in lakhs, the count is made possible by diluting the sample of the blood with diluting fluid before counting and subsequently multiplying the count with the dilution factor. So this is the principle of today's practical of the platelet count. Which are the instruments we are using for the practical of platelet count? Here we are using 1% ammonium oxalate and it is as a platelet diluting fluid. So this is the 1% ammonium oxalate fluid as a platelet diluting fluid, improved Newbar's counting chamber, pricking apparatus which includes lancet, cotton, spirit and cover slips, compound microscope, test tube and test tube stand. So here it is the test tube and test tube stand, RBC pipette, petri dish. So this one it is the petri dish, distilled water and filter paper. So all these are the apparatus which are required for the practical of platelet count. Now here why we are using 1% ammonium oxalate solution as a platelet diluting fluid? Because it destroys RBCs, it acts as an anticoagulant and it preserves platelet. So these are the functions of this 1% of the ammonium oxalate solution. Now we begins with the procedure part. Take platelet diluting fluid 1% of the ammonium oxalate in a test tube. Under aseptic precautions, prick your finger and suck the blood up to 0.5 mark in the RBC pipette. So here it is the 0.5 mark of the RBC pipette and you have to suck the blood up to this marking. Immediately dip the tip of the RBC pipette in platelet diluting fluid 1% ammonium oxalate and suck fluid up to 101 mark. Once you collected the blood up to 0.5 marking, after that immediately you have to dip the tip of the RBC pipette into RBC diluting fluid and suck the fluid up to 101 mark of the RBC pipette. Mix blood with diluting fluid, hold pipette horizontally, wait for 10 to 15 minutes. Why we have to wait for 10 to 15 minutes? Because during this time RBC hemolysis takes place or the destruction of RBC takes place, because here we are doing platelet count practical. At the end of 10 to 15 minutes, discard first 1 to 2 drops of the fluid from the pipette, as the stem of this pipette up to the 1 mark it only contains diluting fluid but not any blood cells. That's why we have to discard first 1 to 2 drops of the fluid from the pipette. Put cover slip over the improved Newbar's counting chamber and charge the smear as you are seeing in this video. Take petri dish, put a filter paper at the bottom of it and add drops of distilled water so it will create moist environment. Now place charge improved Newbar's counting chamber in moist petri dish for 20 to 30 minutes. During this time platelets will settle down. So you have to put this charge improved Newbar's counting chamber in a petri dish where the moist environment is there, as there is a wet filter paper. And after that you have to wait for 20 to 30 minutes, because during this time platelets will settle down. After 20 to 30 minutes, put charge chamber on mechanical stage of the microscope and count platelets in 25 RBC squares. Now you already know the counting area and their squares. So here the center square is for the RBC counting area, and in that square there are number of 25 squares are there, and in every square you have to count the platelets in a 45x lens or high power lens. How to identify platelets? So these are highly refractile round bodies, very small, 2 to 4 micrometer in diameter, well separated from other cells. So whatever the cell you are looking in this photograph, these all are the platelets. These are the highly refractile round bodies, because whenever we try to do the fine adjustment, the platelet will appear and disappear. That's how we are able to find which is the platelet and which is some type of dust material and some type of other cell. So in this photograph, whatever this type of cells you are seeing, these all are the platelets. So you have to count these platelets in every 25 squares of the RBC area. Now the calculations. Calculation of dilution factor. Dilution factor is equal to final volume achieved divided by actual volume taken. So final volume achieved is equal to 101 minus 1 is equal to 100. The final volume achieved, it is the 101. But here the whatever the number 1 mark of the fluid, we are not taking into the this dilution part of the blood. That's why we are not including this markings of the RBC pipette, and here we will get only the final volume achieved, which is the 100. Actual volume taken 0.5, as we collected the blood of the up to the 0.5 marking. So 100 divided by 0.5 is equal to 200. So dilution factor of this practical is 200. Now final calculations. Here we have to find out the area of RBC square. So this is the 3 by 3 mm square. The whole of the one counting area of this improved Neubauer's counting chamber having the 3 by 3 mm square. 3 by 3 mm square means 3 mm in length and 3 mm in width. So here it is the area of RBC square. That means RBC square, it is of the 1 by 1 mm. Alright. So here the area of RBC square is equal to 1 mm multiplied by 1 mm is equal to 1 mm square. Here the depth 1 by 10 mm. Depth means it is the gap between the cover slip and the counting area. So volume of RBC square is equal to 1 mm square multiplied by 1 by 10 mm is equal to 1 by 10 mm cube. So this is 1 by 10 mm cube. It is the volume of this RBC square. Now 1 by 10 mm cube contains n number of platelets. Then 1 mm cube contains how many number of platelets? So 1 mm cube contains 1 multiplied by n divided by 1 by 10 is equal to n multiplied by 10 number of platelets in 1 cubic mm. Now as per our principle, we have to multiply the count with the dilution factor. As our principle said that as normal platelet count runs in lakhs, the count which is made possible by diluting the sample of the blood with diluting fluid before counting and subsequently multiplying the count with the dilution factor. So whatever the count we will get, we have to multiply it with the dilution factor. So our dilution factor is 200. So we have to multiply this n multiplied by 10 multiplied by 200 is equal to n multiplied by 2000. So this is the final formula for the calculation of platelet count. So whatever the number of platelets you will find in this 25 RBC squares, just add it, and that's how you will get the n value. Just putting n value over here and it multiplied by 2000, you will get your platelet count. So this is how you have to do the calculations for the platelet count practical. Functions of platelets. Role in hemostasis. Hemostasis means spontaneous arrest or stoppage of bleeding. Role in clot formation, role in clot retraction, role in repair of injured vessels, role in defense, a little bit there, but still it's not a function of platelets, it's purely a function of WBCs. Transport and storage function. So all these are the functions of platelets. Properties of platelets. Adhesiveness means stickiness, aggregation and agglutination. Normal count and variations. Normal platelet count 1,50,000 to 4 lakh platelets per cubic mm of the blood, or in some other way 1.5 lakhs to 4 lakhs platelets per cubic mm of the blood. The whole unit you must have to speak. Without the unit the number is nothing. Physiological variation, that means platelet count may vary according to the age, sex, after meal, after severe exercise, at the high altitude. Now the variation of the platelet count, whenever the platelet count below the normal, the condition is known as thrombocytopenia, and whenever the platelet count above the normal, the condition is known as thrombocytosis. So thrombocytosis, increase in the platelet count, and the conditions, for example after trauma and splenectomy, in these conditions there will be the increase in the platelet count, and the condition is known as thrombocytosis. Now thrombocytopenia, that means decrease in platelet count less than 1.5 lakhs, and the conditions are the hypersplenism, viral infections like dengue, and bone marrow depression. So all these are the conditions in which there is a decrease in the platelet count below the normal values, and the condition is known as thrombocytopenia. I hope via this presentation now everyone able to understand how to do the practical of platelet count. If you like this presentation, please try to share it with your friends, group, colleagues and batch.